Assalamualaikum my dear students, welcome to STUDYHIGH.Pk and subscribe keeping contact for more videos update. Today we are moving towards the last lecture of this chapter Carbohydrates. Lecture or topic is polysaccharides and their digestion in human body. First of all, I want to inform you or discuss what are the learning outcomes, what are the important concepts or processes that I am going to putting down into your minds in this lecture. Like we will discuss the structure and properties of starches, different types of polysaccharides, glycogen, inulin, pectin, chitin, cellulose, and the different type of concepts or their roles in human body, or the different type of reactions, their roles in causing different type of diseases or in controlling them. Like why amylose is difficult to digest than amylopectin, or why a diet that consists of high concentration of amylose is difficult to digest as compared to the diet that consists of high concentration of amylopectin. We will discuss it in detail. What is glycemic index and how it helpful in treating the diseases? What is glycogen, how it involved in formation of glycogen molecules, synthesis and breakdown of glycogen, how inulin is used in determination of glomerular filtration rate, structure and physiology of cellulose, pectin, etc. and different type of concepts that you will become to know after completion of my lecture. Like why amylose exists in helical structure, why plant stores much of the energy in the form of amylose, etc. Now we are going to start the first topic or the first polysaccharide of this lecture, that is starch. What are the sources of the starch? Cereals, potato, legumes, etc. Like here are the potatoes, cereal crops. These are the major sources of starch, and it is the most abundant carbohydrate that is being used by human beings as a source of diet. It has two types, amylose, amylopectin. Its importance that we are going to discuss you. The most important thing that you must know, that starch is a polymer of alpha D glucose that is connected with other glucose through alpha 1,4 glycosidic bond. And here is the glucose molecule. It is in alpha configuration. Why? Because this hydroxyl group is projected downward and this CH2OH group that is furthest away from the aromatic carbon is projected upward. That's why this is an alpha glucose. It is connected with the second glucose monomer through alpha 1,4 glycosidic bond. And here is the simplest structure of amylose, in which a number of glucose molecules connected with each other through alpha 1,4 glycosidic bond. This is the structure of amylose, and it also exists in helical form like this. Why this exists? If you know about the structure of helical structure of different types of proteins, like because of hydrogen bonding between the one amino acid and another amino acid, some proteins exist in the form of alpha helix. Similarly, such hydrogen bonding also exists between the glucose molecules that involve in formation of amylose. Like carbon 2, the hydroxyl group connected with the carbon 2 of one glucose can form a hydrogen bond with the carbon 3 of the next glucose. In this way, this hydrogen bonding causes the amylose to exist in helical form. As a result of this helical structure, amylose exists in much more compact form as compared to the amylopectin, which exists in branch form. Like this is the amylose molecule. This is also present in alpha helix or helical structure. It is more compact as compared to the other polysaccharide or amylopectin. Like amylopectin consists of a branch. Like here is the branch. Just think, this is another glucose monomer that is branching the linear unbranched chain of amylose. So amylose also exists in branch form and consists of alpha 1,4 and alpha 1,6 glycosidic bond, and it is less compact as compared to amylose. And amylose is easy to crystalline, as easily to go into crystalline form as compared to the amylopectin. That's why amylose, because of its crystalline nature, or easily go into crystalline form. Because of its compact structure due to the formation of helical structure, it is difficult to digest as compared to amylopectin, which exists in branch form. Whereas, because of its compact structure, amylose also used by the plant to be used as a transport or storage form because it is a compact form, so much more of the starch that is being used by the plant as a storage is amylose form. Myelopectin is also, but much more concentration is of amylose. So we have discussed you the importance of helical structure, why it formed because of the hydrogen bonding. Similar hydrogen bonding that converts the protein into a helical form causes the amylose to exist in helical structure, and it is much more compact. That's why it has a different type of roles, and it is difficult to digest in human body because it is a compact and it is easily crystalline or can easily go into crystalline form as compared to the amylopectin. Now we are going to discuss the much more important concept of this topic lecture, glycemic index. It is a number from 0 to 100 assigned to any food. Like if a food that a person eat, just think this is a person that takes in a food, and after 30 hours, after 30 minutes or 1 hour, the food that he taken in will be broken down and will be entered in our bloodstream. So then you will check the what is the glucose level of the in the blood. So according to the glucose level that this food can be raised, we give a number from 0 to 100. If this glucose or this food generates or causes the maximum increase in blood glucose level, this will arbitrarily given the number 100. If this person takes in cellulose, inulin that has no nutrition value, that does not digested by a human body, and that does not cause the increase in blood glucose level, so they have a number that assigned is 0. In this way, we different type of foods we assign a definite number to different type of foods on the basis of how much blood glucose level they can raise. Likewise, the importance of this glycemic index is that they show that a food that has a maximum glycemic index shows that our body can rapidly or easily digest this food, and our body's response of here is insulin. So here is a graph that shows the blood glucose level after eating a food by a person in 15 minutes, 30 minutes, 45 minutes, 60 minutes. After 50 minutes, the blood glucose levels start rising, like here 5 mole per milliliter. After 30 minutes, you found a maximum blood glucose level. Then the blood glucose level further going down. Why? It shows that the glucose or the food which raises the glucose in the blood is start moving into some other form or being used by the body, or insulin causes this glucose to be converted into glycogen form or other form in the body. It shows this. So on the basis of the graph of glycemic index, you can also observe or interpret the insulin response to the blood glucose level. If insulin is not responding, this graph will be going down in this direction or straight. In this way, glycemic index will help you in treating different type of diseases. Like if a person is obese, just think this person is suffering from obesity. Here is a person that is suffering from obesity and is very upset on this on obese nature or overweight. You will suggest a diet to this person that has low glycemic index, like saturated fatty acids, rather than a diet that has high glycemic index rather than carbohydrate. So if you know the glycemic index of different type of foods, you can suggest, and you are doctors, you can suggest you must need this diet and you must be avoid this diet. In this way you can treat the diabetes obesity of this patient. Likewise, a patient who is suffering from diabetes mellitus and has high blood glucose level in the blood, high blood glucose level like diabetes patient, you should suggest the diet that or food that has low glycemic index that will cause low increase in blood glucose level after digestion by human body. In this way, glycemic index help you in treating different type of diseases, in controlling body weight, in controlling diabetes, mellitus, etc. Let's move towards our next topic, glycogen. Glycogen, physiological difference of glycogen in liver and muscles. How insulin initiate its formation. Why muscle do not increase blood glucose level. These are the important concepts that we are going to discuss. First, what happens? Basic thing, then the other process we will discuss. Insulin is a hormone secreted by our pancreas. It, through our blood circulation system, portal system, enters the liver. After entering into our liver, it causes the hepatocytes, the cells of the liver, and which causes the activation of different type of enzymes that involve in synthesis of glycogen. So this is a simple process. Now I am going to discuss in detail what in really happens in the human body. This is the structure of glycogen. It also consists of alpha 1,4 and alpha 1,6 glycosidic bonds like those happen in myelopectin. But it is much more branched than that of myelopectin. But there are other differences, like amylose formed in plants, whereas glycogen occurs in human beings. It is much more branched. It has different type of functions and different type of origin. That's why glycogen is also called as animal starch. Like plants store carbohydrate in the form of starch, animals store carbohydrate in the form of glycogen. That's why we also call it animal starch. Now I am going to show you how glycogen is synthesized or stored in human body. Here is a basic process that really happens. Here is a blood glucose. As you can see, this diagram is exactly a copy here of this. Here is the glucose in our blood that is converted by the glucokinase in liver. You must know that in liver, glucose is converted into glucose 6-phosphate by glucokinase, whereas in muscles, the glucose is converted into glucose 6-phosphate by hexokinase. These are the two different types of enzymes on the basis of glucokinase causes the conversion of glucose into glucose 6-phosphate. In muscles, it is a hexokinase that causes the conversion of glucose into glucose 6-phosphate. So you must know about it also. Glucose 6-phosphate. It is converted into glucose 1-phosphate by an enzyme phosphoglucomutase. So this glucose 6-phosphate has been converted into glucose 1-phosphate. This glucose 1-phosphate reacts with the uridine triphosphate and got converted into uridine diphosphate glucose. 2-phosphate has been emitted in the form of pyrophosphate, and glucose 1-phosphate has been converted into uridine diphosphate glucose. Here, an enzyme known as glycogen. It consists of 2 tyrosine, which are amino acid. It binds one glucose molecule, second, third, fourth. So glycogen, here is the glycogen protein. It binds at least 8 glucose monomers, not glucose, UDP glucose, uridine diphosphate glucose. When it binds at least 8 glucose molecule, then another enzymes enters glycogen synthase. It takes the control further and add the further glucose molecules, glucose residues, formation of a number of 1,4 glucosyl units through alpha 1,4 glycosidic bond. A number of glucose monomers has been attached. Then the branching enzyme also enters and causes the branching at different sites of the glucose chains causing the formation of glycogen. In this way, this is a process that is basically happening. We are not going to discuss in detail in this lecture because glycogen has a separate chapter which we will discuss in second year. But I want to show you that how this process is happening in human body. This glucose converted into glucose 6-phosphate, glucose 1-phosphate, uridine diphosphate glucose by using an enzyme glycogen that form a primer for the glycogen synthesis. What is the primer for glycogen synthesis? Its primer is a chain of at least 8 glucose monomers. This glycogen synthesis then further causes the elongation of the chain of glucose that being connected through the alpha 1,4 glycosidic bond. Then another enzyme, branching enzyme, that cause the branching of that chain at different sites. Now, this is the glycogen molecule that is being stored in our liver. If you want to convert it into its original form, glucose form, it is being converted by a glycogen phosphorylase that cause the breakdown of one glucose residue, then second, third, and different type of enzyme, deep branching enzyme. So one is a branching enzyme that involves in formation of glycogen. Here will be a deep branching enzyme, glucose transferase. So as a result, there will be a formation of glucose 1-phosphate. This glucose 1-phosphate will be converted into glucose 6-phosphate by an enzyme. This glucose 6-phosphate has a different role in liver and muscles. Like in muscles, this glucose 6-phosphate is used by its muscle own cells through a process of glycolysis for the production of a different type of ATP and other type of energy sources. Whereas this does not happen in liver. Liver also contains glucose 6-phosphatase that converted this glucose 6-phosphate into glucose that enters in our bloodstream. Whereas muscles lack this hormone, glucose 6-phosphatase. As a result, they does not increase the blood glucose level, but they use this glucose 6-phosphate through glycolysis and different types of cycle like Krebs cycle for the production of energy. This is the basic reason. Because of lacking of glucose 6-phosphatase, muscles do not increase the blood glucose level. So they use it only for their own cells, for the production. So whereas what happens in liver? Liver consists of glucose 6-phosphatase. It causes the conversion of glucose 6-phosphate into glucose. Now this form can enter in our blood and can be transported to the other tissues like kidney and other brain. Our brain, liver can convert glucose 6-phosphate into glucose that can be transported to the other organs of the body as well that need glucose. Now we are going why muscle do not increase blood glucose level because they lack an enzyme glycogen phosphatase. How insulin initiate the enzymes glycogen and another enzyme glycogen synthase. And what is the glycogen physiology in difference of glycogen physiology? Liver after breakdown of glycogen causes the increase in blood glucose level that can be transported to the other organs of the body as well. Whereas in muscles, the after breakdown of glycogen, glucose does not increase or the muscle does not cause the increase in blood glucose level. They use the glucose 6-phosphate for its own cells only. So this is the basic difference between the formation and breakdown of glycogen that occurring in muscle and liver. Whereas, and one other difference, glucokinase is an enzyme that causes the conversion of glucose into glucose 6-phosphate in liver. Whereas hexokinase is an enzyme that is present in muscles and involved in conversion of glucose into glucose 6-phosphate. To discuss inulin, sources of inulin. Here are the different type of sources of inulin like artichokes, dandelions, these are the two dandelions, dahlias, dahlia after the name Andrew Dallas, after the name of a scientist. So these are the two. So we are going to discuss the much more important topic of inulin, that is structure and how it involved in determination of glomerular filtration. Like here is the structure of inulin. Inulin is a polymer of beta 2,1 fructose. Beta fructose. It is a polymer of fructose, whereas starch, glycogen, cellulose are the polymer of glucose. So inulin is a polymer of what? Fructose. One fructose connected with the other through beta 2,1 glycosidic bond. This is the NMDCAT carbon glycosidic bond that is projected onward. Now we are determining whether it is in alpha glycosidic bond or beta glycosidic bond. This is the carbon that is furthest away from the NMDCAT carbon. It is also in downward direction, projected downward. Both are in cis form. So this is a beta fructose according to the rules which I already explained you in previous lectures. So this is a beta fructose. This is a carbon 2 and formed a glycosidic bond with carbon 1 of the next fructose. So beta 2,1 is the bond that exists between the different fructose monomers of inulin. Some books like Harper's Illustrated Biochemistry has mistakenly gives some wrong diagrams of different type of disaccharides and polysaccharides as well. So you must take care of these things as well. Like here is another inulin molecule. It is connected with beta 2,1 glycosidic bond between different type of fructose monomers. So this is the structure of inulin. You must know about it as well. We are going to discuss how inulin can be used in determination of glomerular filtration rate of a patient to check whether a patient's kidney are working properly or not. For this, we inject a definite concentration of inulin into the bloodstream through IV. So we determine how much this concentration can raise, or we raise the blood plasma concentration of the inulin of the patient to a definite level. What will happen? This inulin will pass it into the kidney, will pass it through the kidney because our whole blood passes through the kidney for filtration. This will also be filtered by the glomerulus Bowman's capsule. It will also pass into the tubular system and will be excreted in the urine. So what are the important characteristics of this inulin that make it a useful or a gold standard method that used to determine whether a kidney is functioning properly or not? Because inulin is soluble. It is soluble in aqueous solution, whereas cellulose is non-soluble, and that's why inulin can be used. Second, it has no nutrition value. So once it passes through the glomerular filtration, Bowman's capsule, it will not be reabsorbed by our renal system. So the compound that must be used in determination of glomerular filtration rate should not be absorbed. It should be excreted. And on the basis of the concentration of inulin present in the urine and what the concentration we have injected into the human body, in determining this concentration, we can check how much extra insulin we injected to the patient and how much we receive in the urine. On the basis of these things, these statistics, we can determining whether our kidney is filtering the waste material properly or not, or at what extent it is filtering and at what it should be filtered. So inulin is used for the determination of glomerular filtration rate of different patients and helpful in determining whether a kidney is filtering properly or not. So in this way inulin can be used. Now we are going to discuss some much more important thing: cellulose, chitin and pectin. First I want to show you that what is the structure of cellulose, chitin and pectin, because these are much more not much more important as compared to inulin or cellulose, glycogen, starch. But you must know, in some cases you need cellulose. You must know cellulose is the glucose polymer of beta 1,4 glycosidic bond. Like a number of beta 1,4 glycosidic bonds, beta glucose connected with beta 1,4-O glycosidic bonds to form the cellulose molecule. Whereas in chitin, there are N-acetyl glucosamine. Chitin is a polymer of N-acetyl glucosamine and consists of beta 1,4 glycosidic bond again. Whereas pectin is a polymer of galactouronic acid and consists or linked with alpha 1,4 glycosidic bond. I want to show you a little example how these things are occurring in human. If you remember my first lecture of isomerism, you must know that glucose can be exist in different type of forms. Alpha form, if this CH2H group is projected upward and the hydroxyl group is projected downward, it will be alpha glucose. But in beta glucose, that involve in formation of cellulose molecule, this hydroxyl group is also projected upward. Both are in cis form. That's why this glucose is beta D glucose. And in this, it forms a beta glycosidic bond with carbon 4 of the next glucose. So in this way, it involves in formation of glycosidic bond. But what will happen if this CH2OH group or this hydroxyl group, it has been changed or in glucosamine, this hydroxyl group has been converted by NH3 positive. This is the structure of glucosamine. That may be alpha D glucosamine and beta D glucosamine. This is the structure of beta D glucosamine because hydroxyl group and CH2OH group are in cis form. But what happens in chitin? In chitin, we have converted this NH2 into N-acetyl glucosamine, CH3. So the glucose that involved in formation of chitin consists of this N-acetyl glucosamine along with the glucose. This is the basic monomer that involved in formation of what? Chitin consists of galactouronic acid. If this is the structure of glucose, right? Its carbon 6 that consists of CH2OH group has been oxygenated through a process of oxidation, got converted into COOH, glucose into glucuronic acid. We convert the carbon 6 CH2OH group or oxidize this carbon into COOH, carboxylic group. So it is converted into glucuronic acid. But the monomer of pectin is galactouronic acid. It has OH group projected upward on carbon 4, not downward. This is pectin. It consists of galactouronic acid because the hydroxyl group that is present on carbon 4 is projected upward. So this is the basic difference between the glucose and galactose. In glucose monomers, the carbon 4 is projected downward, as you can see in cellulose, as you can see in chitin, whereas in pectin, it consists of galactouronic acid. So you must remember what structure you are going to drawing or what is the monomer of pectin. Pectin consists of galactouronic acid. In carbon 4, how its glycosidic bond is also different. It is in this direction, whereas the glycosidic bond of chitin and cellulose is in this direction. So this is the difference between the glycosidic bonds are present in pectin and other polysaccharide. Likewise, its carbon CH2OH group as present in group. So this is also a difference in pectin and other structure. Pectin also consists of some acyl, some methyl substituted groups in some of its monomers. So this is the structure of cellulose, chitin, pectin. You must know the base structure of the monomer that involve in formation of these structures. Now I am going to solve some MC. What is the major difference between cellulose and pectin? Cellulose and inulin. What is the physiological difference between cellulose and inulin? Both are the polysaccharide. One is a polysaccharide of beta D glucose. Inulin is a polysaccharide of fructose. So what is the difference? Solubility, nutrition value. Both has no nutrition value. Both does not reduce the blood glucose level on their digestion. But though both could not be digested by human body. So both has no nutrition value. Second, solubility. Solubility is the basic difference between these two molecules. Why? Inulin is soluble, whereas cellulose is insoluble. So you must know the properties of different type of polysaccharide that can be asked in your professional exams. Question number two. Which of the following is a structurally acidic polysaccharide? Cellulose, glycogen, inulin, pectin. As I have explained you, in inulin, the carbon 6 of galactose has been carboxylated or has been oxidized into COOH. So pectin is a structurally acidic polysaccharide and it consists of galactouronic acid, not glucouronic acid, because on carbon 4, as you know, galactose and glucose are the epimers, C4 epimers of each other. In galactose, COH group is projected upward, in glucose projected downward. So these are the some important discussions, concepts that we have discussed today. We discussed structure of cellulose, pectin and chitin. We have also discussed how structure of inulin consists of beta 2 1 glycosidic bond and how inulin is involved in formation of or germination of glomerular filtration. Sources of inulin, dandelions, artichokes, dahlias. We have also discussed that glycogen is sensitive in human body, like different type of processes that occurring in liver and muscles. We have also discussed glycemic index, how it helpful in treating the obesity, overweight, and diabetes patient. What type of diet you must suggest? Diet with low glycemic index and starch sources. Why myelopectin is difficult to digest as compared to some other polysaccharide etc. In next lecture we will discuss glycose aminoglycans. We are going to start our next chapter that consists of GAGs and glycoproteins. So hope you best of luck, best wishes, like, subscribe, share. Thank you, Allah Hafiz.